Hello all, welcome to Renan's PhD defense. And this is one of the most important day for my academic career because he is my first PhD students. If you don't know what is the life of assistant professor, we do a lot of different things and it become messy, and then we have to build a lab and do all that other things now. So it's very critical. The first PhD students has to be, that organized and very well to bear us and do the work. So Renan, I could not think of any better students than him to start the lab with him. So he is my colleague that we grow together, and he did so well. Not only he enabled his PhD work so well, but also he enabled other students that follows. So he set up the lab. He did amazing work despite pandemic. So I'm super proud of him. Without wasting any time, you don't have to trust me. Just see what he has done. So this is all Renan now. If you can take the mic and present your work.
Thank you, Sanjay. Thank you for the introduction. And thank you, everybody that are here watching my presentation. My name is Renan. I'm a fourth-year PhD student here at the UCLA. And today I'll be presenting or defending my PhD thesis that is titled Resilience of Stormwater Treatment Systems Under Changing Climates. I would like first to thank the committee, Dr. Mohanty, for all of your guidance throughout my PhD. And
so I just lost the audio.
Yeah, I can't hear anything either.
I lost audio too.
Okay.
am I back?
Yes.
Yes.
Good.
All right, perfect. Sorry about that. So thank you, Dr. Mohanty, for all of your guidance throughout my PhD. Thank you also, everyone in the committee, Dr. Mahendra, Dr. Ambrose, and Dr. Jasby. I wish we had more time together, but know that I've learned a lot from you indirectly by talking to your PhD students and postdocs. So thank you so much for your direct and indirect guidance throughout my PhD. I would also like to thank everyone that is watching on Zoom, friends and family from pretty much all over the world. And if you permit me, I just want to say a few words in Portuguese because I know there are my parents in Brazil are watching. Obrigado, pai, mãe, irmão, todos os amigos que estão assistindo do Brasil. Muito obrigado por todo o suporte durante o meu doutorado e obrigado por estar aqui assistindo. And I also want to say a few words in Italian because there may be some Italians online. Grazie a tutti per guardare questa presentazione. Se avete qualsiasi domanda potete scrivere sul chat o chiedete al fine della presentazione in inglese ovviamente, ma grazie mille per essere qui. All right, so let's get started. So my presentation, I'm a visual learner and I like to see where I am at and where I want to go to. So I always want to start with a presentation outline. So first in this presentation, I'll start with my journey as a researcher here at UCLA since 2017. And then I'm going to get a little bit more into the science side and talk about the resiliency of stormwater treatment systems and break it down into three specific areas. First, present about the causes of performance variability in stormwater treatment systems, which are chapter 2 and 3 of my PhD thesis. Then I'm going to get into the quantification of performance variability of those systems, which are chapter 4 and 5. And then lastly, I'm going to talk about the mitigation strategies to reduce the performance variability, which is chapter 6. I'll then finish with a few recommendations on for future studies, and also talk a little bit about my next career steps. All right, so first, my journey as a researcher here at UCLA. It has been a long journey, but definitely very fun and enjoyable. I really enjoy every single moment of my research here. The picture to the left is actually the first day that I got into the lab or into the grad office. It's a different grad office now. We were actually at where Dr. Jasby's graduate students are at now. And I've had a lot of, dealing with troubleshooting equipment, ion chromatograph specifically, the picture in the middle, and I was doing some troubleshooting there and also reading some puppy training books, because along my journey, I also got engaged and got married and got a puppy. So lots of excitement there. And during the engagement, actually, my lab group helped me plan everything. So thank you everyone who helped me on that. It was some extra duties through the laboratory. And I also did a lot of work in the laboratory, troubleshooting experiments. But not just inside the laboratory, I also worked outside. Did a lot of sample collection at the stormwater and sediments or wildfire residues, mostly at the Ballona Creek, as well as the Ballona wetlands, which are located in Los Angeles. And also the picture in the middle, it was a laboratory experiment that I did at the top of Bolter Hall, the engineering building here at UCLA. And it was definitely the best view of any experiment that I've ever had, because I was pretty much looking at Beverly Hills and doing experiments with bacteria. But not everything is fun in the lab. And I obviously had many failures, which for me, it was just a boost to keep going. We've done a lot of experiments with electrochemical biofilters, which failed, as well as fungal augmented biofilters. And those two, we've learned a lot. There's a lot of opportunity there. But those were definitely like a lot of time spent and definitely some progression, but we could not conclude those experiments. But the picture in the video is the one that I want to highlight. That's a Petri dish, and it's called PBS, which stands for phosphate buffer solution. And the problem with that picture is that whenever you have that phosphate buffer solution, you don't want to see any dots in those Petri dishes. And as you can see, there are many dots there. The problem with that is that I lost all of my data from that day, 100+ measurements. And I remember taking this picture and then looking at my phone, and there is a sign right above the PBS, which is the exit sign. And for me, I don't know if it was a sign or a indirect sign of giving up, but I'm a person who never gives up. I always figure it out things. Even though that exit sign was there, I just kept going. And when I was 2.5 years into my PhD, just in my prime time, getting everything done, having many experiments lined up, we had the COVID pandemic. So the laboratory, or my home office, became my laboratory. But as I said, I never give up, I keep going. And I found in data science a really good opportunity to apply towards stormwater treatment systems, use online databases, peer review articles, as well as other data that we had produced in the lab to keep going. So as a summary of my journey, I've published 13 articles so far, where 4 of them are lead authors, and I have 4 more other articles in preparation. I also authored a book chapter on biochar and how biochar can help the resiliency of stormwater treatment systems. I taught 3 engineering disciplines here at UCLA, and I really enjoy, and it helps me with my communication skills and getting to know other undergrads and expose them to research as well. I also worked as a consultant for multiple companies and institutions through UCLA as well as privately. And I'm currently a fellow at the National Science Foundation Infuse program, where I work with food, energy, in the food, energy, and water nexus, and also a fellow for the Los Angeles Urban Center, where I work with wildfire-related studies. And along the way, I've received many fellowships and scholarships that have been supporting my research. All right, so let's get into the fun stuff, or the fun science, and talk a little bit about the resilience of stormwater treatment systems. But before we get too deep, let's look at the big picture here. My PhD is pretty much about water science, right? The big picture. And why is it important? So if we look at this image to the left, when we think of how the water moves through the earth, we have the evaporation of water from water resources and forests, and it forms these clouds that move along, and there is a precipitation that either the water moves downward and infiltrate through the Earth's subsurface, or it becomes what we call the stormwater runoff. This stormwater runoff can carry contaminants and pollute water resources. So if we take care of this stormwater runoff, we have a very good opportunity to make sure that we protect these water resources, either groundwater or surface water. But how do we protect these water resources? Well, we use stormwater treatment systems, right? Pretty much the stormwater runoff, it can run into the stormwater treatment system, which hopefully is going to clean and remove contaminants and then protect the water resources. So in this picture to the left, we have 4 examples of stormwater treatment systems. The first one is a retention pond. Imagine as a big pool of water, where water comes in, stormwater comes in, it stays inside for a long time, and contaminants are degraded for days, and then water flows out. The second one, the top right picture, is a grass swale, which is a shallower type of stormwater treatment system, and it's usually built next to roadways, and water passes through it pretty quickly. Some contaminants are removed, some are not, and then water exits. The bottom left one is a media filter, which is another type of stormwater treatment system. Pretty much what we do here is that we replace the soil with sand or some other type of material that has high hydraulic conductivity. So it helps the water infiltrate to another drain, and that water can be discharged somewhere or maybe recharge the groundwater. And the last one, the top right one, or the bottom right one, is a bioretention system, which is pretty much the most complex one. We have, we have a biofilter with plants on top, and we have a media filter. We replace soil with some type of media. And we also create a ponding zone that helps contaminant removal. So at the bottom of it, there is an under drain that can collect water and deliver this water somewhere else. So as you can see, these systems are pretty natural. They are natural engineered systems, and they are usually implemented to remove contaminants and meet some regulations that governments put in place. They can be implemented in urban and agricultural areas, but are they reliable? Are these systems actually reliable into removing contaminants? So short question is, no, they are not. They are unreliable. And this is a paper that we published in 2021. I did a lot of data analysis by collecting field data for contaminants from influent and effluent concentration of contaminants in stormwater treatment systems. And this graph to the right, the x-axis is are many different contaminants, and the y-axis is the concentration of that contaminant. The orange box plot is the concentration of that contaminant in the influent water. The blue box plot is the concentration of that contaminant in the effluent water. What we see is that some contaminants, first, there's a high variability. Some contaminants like the TVS, there is a very likelihood that that contaminant is going to be removed. However, if we look at dissolved metals, more specifically iron, we see that the concentration in the effluent is actually higher than the concentration in the influent. So in that case, that stormwater treatment system is actually a source of contaminants. And nowadays, there is still no guidelines on how to improve the performance of stormwater treatment systems. So before we try to understand how to increase the performance, let's try to understand what is the reason of this variability. So as any natural system like a tree has flowers or leaves during the summer, and it loses its leaves during the winter. And that's because of this climate change. So climate is actually a really big contributor for this performance variability of stormwater treatment systems because they're natural engineered systems. So variables like weather, precipitation, temperature, and even wildfires can affect the performance of the stormwater treatment systems and make them fail in the field. So just to exemplify how this resilience of the stormwater treatment system works, the first graph, it's basically a stability curve by the variables stage. So let's say we have two steady states, and these natural systems working well. When you have a stressor, like a heavy rainfall or a high temperature or low temperature, that's gonna make the state change to a lower stability stage. And that's gonna end up being picture 4A, which is an ecological system with a new steady state that it's unreliable. However, picture 4B, if we have human interventions and we mitigate those problems, we can bring that steady state back to the original steady state and keep those performance going well. So how do we improve this resilience of stormwater treatment system? This is pretty much my research here. First, we understand the causes. What is causing that performance? Is it weather? Is it high rainfall? Is it temperature? And that's going to be chapter 2 and 3. Then I'm going to talk a little bit about the quantification, quantify how much is actually varying the performance, and that's chapter 4 and 5. And last, we're going to propose some mitigation strategies to reduce this performance variability and make sure that these stormwater treatment systems are actually removing contaminants rather than polluting the water. All right, so first let's talk about the causes. What is causing this performance variability? There are 2 chapters in my PhD thesis, chapter 2 and chapter 3. Chapter 2 has been published already at the Water Research Journal, and chapter 3 is currently under review. Because of time, I'm just gonna focus on chapter 2 here. But the big difference between these 2 chapters is first, the contaminant. In chapter 2, I focus on nitrate, which is a specific type of contaminant. Chapter 3, I focus on bacteria. And chapter 2, I focus on 4 different types of stormwater treatment systems, whereas chapter 3, I focus just on one stormwater treatment system. All right, so chapter 2 is about the nitrate removal uncertainty in stormwater control measures or stormwater treatment system. And we were looking at, is it the design or the climate, the culprit for this performance variability? So in the picture to the left, we can see that there are many climate factors that can affect nitrate removal in stormwater treatment systems, such as rainfall intensity, the antecedent drying duration, which is the time between 2 rainfalls, and is it a warm day, is it a cold day, or is it a warm climate or a cold climate? That's outside. There are some design factors inside the stormwater treatment system that can affect, such as the vegetation zone. Do we have plants? Which type of plants? Are they helping the microbiome or not? We also have filter media zone. Which type of filter media do we have there? Just sand? Do we have compost? Do we have other type of media? And also designs like the submerged layer. Do we have water that is always inside, or is it just a empty system all the way, meaning water just passes through and exits? So the objective of this paper was to evaluate the relative importance between climate factors and the design factors on the removal of nitrate. So the methods that we used was pretty much a literature review and use the international BMP database to collect data and do a lot of data analysis to figure it out which one's more important, design or climate. So in this picture here, or these graphs, the y-axis is the different climates based on the Köppen-Geiger classification, and the x-axis is the removal of nitrate. Positive removal means that the system is actually removing nitrate. Negative removal means that the system is actually a source of nitrate, so it's polluting water resources. So far, when we look at these 4 different types of stormwater treatment systems, media filter and grass soils are not indicated to remove nitrate. They are actually most of the time the source of nitrate. So if you want to implement some stormwater treatment system to remove nitrate, do not use those 2. When we look at retention pond and bioretention systems, they mostly remove nitrate, but they also export some nitrate. So what's the reason behind that? So when we look a little bit more closely, the picture to the left is the data for bioretention systems that are located within the same climate. And we see, and we were comparing if the presence or absence of plants would impact the removal of nitrate and other nutrients. And what we saw is that it doesn't really affect. And that's because of bioretention systems, they can also have compost that has a lot of nutrients. And then you're not really going to remove just based on plants because there's a lot of nutrient leaching. The picture to the right, the graph to the right is about retention ponds. The most important part here is that the average pond depth is actually very important. So the shallower the pond, the more nitrate you're going to remove. And we explain those results based on, to remove nitrate, you need a denitrification zone, which is usually at the very bottom of the pond because it's the most anoxic zone. So the shallower the pond, the more likely the nitrate coming in is going to reach that zone. Whereas when you have a very big pond or a very deep pond, you're not going to have that mechanism happening. So a few takeaway messages. Retention ponds are definitely the best stormwater treatment system to remove nitrate. And media filter are counterindicated because they mostly export nitrate, despite of the climate that they are located. Bioretention ponds or bioretention systems, they're highly unreliable. And to improve that reliability, we can add some amendments like iron-based media or media mixtures to increase that nitrate removal and make them more reliable. All right, so now that we understood how some of the causes like climate and design, it's time to quantify them. How much is this variability actually taking place? This is chapter 4 of my PhD thesis, and it has been published at the Water Research Journal previously. And here I'm looking at how wildfires can affect the microbial quality of water resources. So let's look at the big picture. Here to the left, we imagine that we have a wildfire somewhere. We have a lot of trees burning, maybe houses, hopefully not. And after that wildfire, you have a lot of residues that are left behind. And at some point, you're gonna have a rainfall sooner or later. And what happens is that this rainfall is either gonna move those wildfire residues through the subsurface down to the groundwater, or it's gonna move those wildfire residues along the surface of the earth as a stormwater runoff and deposit them at surface waters. But what is gonna happen to the microbial quality of that surface water because of the presence of these wildfire residues? Are they gonna help bacteria grow? Are they gonna kill bacteria? It was unknown. So we answered that question. So the objective is to examine the effect of wildfire particles on the fate and transport of pathogens. So our first hypothesis was that microbial transport through subsurface would increase in the presence of particles, but transport would depend on particle type. When we looked at the subsurface transport, we saw that there is no particle attached facilitated transport of bacteria between bacteria and wildfire residues. So I'm not going to be presenting that data because we disproved that hypothesis. The second hypothesis is that wildfire residues would decrease the viability of pathogens in surface waters. And that's what I'm going to be presenting here. So just some of the methods. We collected stormwater from the Balona Creek. We also collected different types of particles, either burned particles or unburned particles from the Malibu or Woolsey fire, and also used E. coli K12 as our indicator bacteria. Pretty much we mix all of this together into a batch experiment, set it in the incubator for optimal conditions, and then we use spectrophotometer techniques as well as spread plating techniques to measure either the concentration of particles or the concentration of E. coli with time. And of course, each batch had a different type of particles. So what we saw is that this graph, the y-axis is the concentration of E. coli, and the x-axis is the duration of the experiment. So when the top left graph is when you have pretty much water with no particles, just the bacteria. So what we see is that the bacteria grow, stays in their stationary phase, and then starts their die-off. Similar trend is found when we have unburned soil, which is the graph with the yellow dots. So bacteria grow, stays in their stationary phase for way longer, almost 15 days. But when we have wildfire residues there, what we identify is that that growth curve happens, but it's lower. But also around 11 to 15 days, all the bacteria dies. So what is happening here is that wildfire residues, for some reason, are actually helping bacteria to die. And if you look at the microbial quality of a water resource, that's a good thing because you don't want indicator bacteria to be there. So some of the takeaway messages is that wildfire residues, they reduce the growth and they accelerated their die-off. That's a good thing. And also we don't need to worry about the subsurface infiltration of wildfire residues carrying bacteria because we disproved that hypothesis. And even though the wildfire residues can kill bacteria, and that would be a good thing, there's wider implications to water resources. What if you actually don't want to kill the bacteria or the microbiome that it's there? That's what we're going to look at in the next chapter, chapter 5. So chapter 5, it's the only experiment that is still, or only paper that is still under preparation. Hopefully we're going to submit it by next month. And pretty much here, what we looked at is the effect of wildfires on wetlands. And this is because wetlands, they are the main natural contributor for methane emission. And methane is a powerful greenhouse gas, meaning they cause climate change and global warming. So when we have wetlands, they are naturally emitting methane, but we don't know if you have a wildfire next to a wetland or even in the wetland, what would happen to the methane emission next? Is it going to increase methane emission, or is it going to decrease because of the deposition of these wildfire residues? So this is what I looked at this research. So again, big picture, we have a wildfire event. Following that event, it's going to be a wildfire or a rainfall, and that rainfall is going to move the wildfire residues to the wetlands. We know that subsurface infiltration is not a thing anymore, so we didn't really look into that. And when these wildfire residues are deposited in the wetland, what is it going to happen to the methanogens or to the methane emission? Is it going to increase or decrease? Because wildfire residues, they are rich in nutrients. They can provide nutrients to bacteria. We also saw that they can also kill bacteria, or at least indicator bacteria. So the objective here was to quantify the methane emission from wetland sediments when those wetlands are exposed to wildfire residues. So our first hypothesis is that the deposition of wildfire residues in wetland sediments will increase methane emission because of this nutrient interaction. And the second is that wildfire residues will increase methanogens by providing bioavailable nutrients or even micronutrients to support those bacteria. So some of the experimental methods, I did both laboratory and field experiments. And in the laboratory experiments, we created an anoxic condition, or a batch with anoxic condition with wetland sediments, different concentrations of wildfire residues, stormwater, as well as acetate as the substrate for methanogens production. And we kept those batch in the incubator, and we would measure methane concentration in the headspace daily, maybe twice per day, and so on. The field experiments are very similar to the batch experiments, but first I didn't purge nitrogen gas through the batch because I was in the field, but they're 1 liter bottles that are filled up with water, and we have a glass wool on top that permits some water exchange between the bottle and the wetland. And I submerged all those bottles, the picture to the right, into the wetland. So they were submerged there for 5 months. And every month I would come and pick up one sample, meaning one bottle, bring to the lab and analyze the microbial community there, or more specifically, methanogens. So first, some of the lab experiments, the results. What we saw, the graph to the left is the cumulative methane emission from these lab experiments. The x-axis is the duration of the experiment. And the biggest takeaway message here is that when we had wildfire residues, which is the gray line, compared to the control, which had no wildfire residues, we have a significantly increase in methane emission. That's almost 56% more methane emission compared to the control. That impact in the real field, in the natural or in the wetlands, is huge. If wetlands are already the main contributors, a 56% increase is substantial when we look at climate change. The graph to the right, we knew that not every concentration of wildfire residues would cause the same impact on methane emission. So we did a concentration based study. The x-axis is the wildfire residues added in percentage. The y-axis is the cumulative methane emission. And what we saw is that the more wildfire residues we add, the more methane emission we have. But anything below 1% by weight, there's not a clear difference on methane emission. So now that we understood what is happening, why is this happening actually? We know that methane is increasing, but why is this happening? So we looked at a few hypotheses and designed a few experiments. The first hypothesis is that it's known that wildfire residues have high silica content. And by leaching the silica content from the wildfire residues, there is a competition between silicon and phosphorus for active sites. And phosphorus can actually compete with DOC. And the end result is that you increase the DOC concentration in the pore water, which facilitates the production of methane. So we designed an experiment to prove that. We did 3 different batch experiments. First one was a control, just wetland sediments with stormwater. The second batch had the control plus the silicon. And the third batch had the control plus the wildfire residues. And that's the results to the right. So the x-axis is the duration, the y-axis is the cumulative methane emission. And what we saw is that first, the yellow dots are the control. Compared to the control, the batches with silicon actually increased their methane emission, but not as much as wildfire residues. So silicon can partially explain the increased methane emission here. So we kept going and tried to think of something else that could explain based on the wildfire residues. And what we saw is that wildfire residues, they can induce the production of reactive oxygen species, including hydrogen peroxide. And the production of this hydrogen peroxide can actually accelerate the degradation of DOC, mainly big DOCs. And what that happens is that it breaks down into smaller ones, which also facilitate methane production. So we looked into that and we designed an experiment without any wildfire residues, but just with hydrogen peroxide. And what we saw, which is the graph to the right, is that when we had hydrogen peroxide, it actually increases the methane emission. We also, of course, did a leaching experiment with the wildfire residues and showed that wildfire residues could leach hydrogen peroxide, pretty much around 60 milligrams per liter for every 5 grams of wildfire residues. So this is more into the geochemistry side. What is happening to the microbiome actually? So what we thought is that the wildfire residues would support methanogens growth by providing kind of a nutrient-rich environment. When we did DNA extractions and we analyzed the MCRA genes, which is the gene expression for methanogens, we saw that the addition of wildfire residues actually decreased the concentration of those MCRA copies in our samples. And that doesn't really bind with our results because we saw an increase in methane emission. But what we saw in the literature is that one, the increase or the concentration of MCRA genes is not correlated with methane emission in many other studies. And also, we saw that a paper showed that an increase in nutrient in the sample can actually decrease MCRA genes. And that's because of the imbalance between methanotrophs, which are the methane consuming bacteria, as well as the between those and the methanogens, which are the methane producing bacteria. So these are mostly the laboratory studies. And we wanted to confront these studies with the field studies to see if what we're seeing in the laboratory would be reproduced in the field. So we did a DNA analysis from our field samples for MCRA genes. And what we saw is that there is a big time difference here. Our laboratory studies were around 20 to 25 days. Our field studies were up to 5 months. So what we saw is that in the field, when we didn't have any wildfire residues, those MCRA genes start decreasing, whereas the bottles with wildfire residues, those MCRA genes remained high. And we explained that into the different conditions that we have in the field. We have glass wool that permits the exchange of mainly micronutrients that can support the MCRA genes or the methanogens inside there. But we still are looking more into that data. So as a takeaway message, or some takeaway message here is that first, wildfire residues clearly increase methane emission, but it depends on how much wildfire residues are deposited into the wetland. Second, the increased methane emission can be partially explained by the increased hydrogen peroxide as well as silicon content. And the last one is that the MCRA genes, they decrease in the short term, in batch experiments, but they actually increase in the long term when, or they remain high when wildfire residues are present. All right, so now that we understood the causes of the performance variability of stormwater treatment systems, and also we quantify how much it's varying, it's good to propose some solutions, right? So here, I'm gonna propose some mitigation strategies that can be implemented in stormwater treatment systems so we don't have this performance variability. This is pretty much chapter 6 of my PhD thesis, and this paper has been already previously published at the Journal of Environmental Engineering. And also, we published a book chapter within the same contact, but mostly looking at biochar and the range of options that you have to increase the resiliency. So for this paper, we partnered with a consulting firm, and they were trying to decide which biochar their client needed to use in the field to remove stormwater or bacteria. They have many different types of biochar, so they didn't know which one made the most sense. For those of you that don't know biochar, biochar is pretty similar to activated carbon, but biochar is 20 times cheaper. So any project that you can implement biochar, your profit margin is going to go up. So here we had 4 different biochars, and we needed to decide which one was the best. So our objective was to identify the biochar properties that had greater influence on the removal of E. coli in biochar sand filters, because we wanted to combine those biochar properties to the removal capacity of that material. The first hypothesis is that the long-term removal would differ than the clean bed removal. The clean bed removal is the very first exposure of stormwater to these biochar sand filters. And this is pretty much at the long term, if you think of all the active sites getting exhausted, most likely your removal is gonna decrease. The second hypothesis is that the removal performance of biochar can be predicted based on biochar properties. So instead of running the experiment again, if you know the biochar properties, you can decide which biochar you can use for your application. So we designed a few laboratory experiments, mostly with a flow-through columns, injecting stormwater from the bottom up and collecting the effluent and measuring bacteria in the effluent. We had, of course, 4 different types of biochar, and we used duplicated filters for the long-term exposure, meaning 70 plus pore volumes of exposure, and triplicated filters for the clean bed removal, which is just the very first 4 pore volumes of removal. So this graph here to the left, the y-axis, the concentration of bacteria or E. coli, and the x-axis, the different types of biochar. The orange box plot is the removal in the clean bed removal, so at the very 4 pore volumes, and the blue box plot is the long-term removal, which is the 70 plus pore volume. So what we saw is that, for an example, naked char, there is no difference between the short-term removal and the long-term removal. However, for the agricultural carbon, we saw a decrease in the removal, which makes sense because of the exhaustion of active sites. So, but for the terrachar and for the rogue char, we actually saw an increase in bacterial removal. And we explained this because basically biochar can create this external biofilm that can help with bacterial removal, not just direct removal, but also the competition between microbes. We then correlated this data with the biochar's properties, which is the graph to the right. And we saw that the ash content at the short-term removal is the worst thing that you can have. So the more ash you have in your biochar, the lower the removal you're gonna have. And that's because ash can create a lot of hydroxyl radicals, which create an electrostatic repulsion between the biochar and the bacteria, which mostly have a negative cell wall, and then that bacteria is not gonna be removed. But at the long term, that ash content becomes less important. And that's because the more water you inject through the filter, the more ash you're gonna leach, the more hydroxyl radicals you're gonna leach, and that becomes just less important. So some takeaway messages is that first, the clean bed removal is very different than the long-term removal, and managers in the field or stormwater managers, they need to be aware of that and make sure that the biochar that they are using actually also continues removing bacteria at the long term. The E. coli removal or bacteria removal is positively correlated with surface area and carbon content, but it is negatively correlated with ash content and volatile matter. So if you can reduce the ash content of your biochar, it's better for the biochar to remove bacteria. And last but not least, there is a model that we developed. It's out there in the paper, and managers in the future, instead of rerunning this entire experiment, they can just use the model to decide which biochar works the best for their experiment or for their application. All right, so some recommendation for future studies. They come from, of course, my failures, because whenever you see a failure, you didn't learn enough. There's still more room to grow. And for me personally, I prefer, not that I prefer, but when you do it once or do it right once, you just learn one way. If you get it wrong and then get it right, you learn two ways. So when you make mistakes or when you have failures, you actually know more rather than when you just go one way. So there is a lot of opportunity with electromodified biofilters, pretty much trying to change the redox conditions of these biofilters to deplete oxygen and create more anoxic conditions. And that's going to favor the removal of certain contaminants like nitrate. We also have opportunities with the fungal augmented, not biofilter, but horizontal subsurface systems. And that's because we spent a year trying to do those experiments with biofilters, and we've realized that fungi would grow just at the top. So we have a very long filter, but there's just fungi at the top. And that's counterproductive when you want to make sure that the fungi is actually removing the contaminants. So in that case, I propose a subsurface system, pretty much similar to a wetland, but not with long, very large ponds. And in this way, you actually have more fungi on top, and water would flow from one end to the other. And that fungi, because it's not too, it is a shallow system, can actually remove the contaminants inside the system. And some next career steps, I've recently accepted a job offer at Exponent. I'll be working as a senior associate there in their environmental and science practice. I'm very, very excited about that opportunity. I've met some of my colleagues and they're amazing people. They're very smart, and I'm very excited to be part of those projects in a more business-driven world. And last but not least, I would like to acknowledge my lab group. Thank you so much, everyone in this picture, as well as people that have already graduated. I could not have completed my PhD if you were not providing me all the support that you did. I would also like to thank specifically my advisor, Dr. Sanjay Mohanty, for helping me along the way. You're a very wise and nice person, and I consider you not just as a mentor, but as a really good friend. And I would also like to thank my wife, Jane, and my dog, Charlie. There are many failures and success along the way, but what makes it very worth is that at the end of the day, I know that they're at home ready to go on adventures, and that just makes everything worth it. And that's it. Thank you very much, and I'm happy to answer any questions.